today we're going to utilize the scientific method to learn about how DNA can be used to identify a new species. And so by the end of this activity, you should be able to extract DNA from a strawberry and understand how that applies to DNA barcoding. So step one in the scientific method is observation. Our observation is that a new species of fish was found in the lake.
The fishing guides are surprised at the Round Lake Resort because they found a new species of fish in the lake. So what species is it? Where did it come from?
Step two in the scientific method is the scientific question. Our scientific question is, how can DNA be used to identify a species?
Once, only experts could identify species with certainty. But today, however, there's a process called DNA barcoding that can be used.
Step three in our scientific method is the hypothesis. So our hypothesis today is, if the DNA is barcoded, then it can be compared to known species.
So the supermarket uses barcodes to identify their products. DNA barcoding is very similar to this. DNA barcoding works because regions of DNA are the same among species members, but they differ from one species to the next. So the standard barcode for nearly all animal species is a section of a gene called CO1. And this gene codes for a protein in the mitochondria. The protein is involved in cellular respiration. In every animal species, mutations in that CO1 gene have changed it in unique ways that allow us to tell one species from another using their DNA barcode.
Step four of the scientific method is experimental design, and this experiment is designed to compare the barcode of a new species to the barcodes of known species.
So step one in DNA barcoding is to extract or remove DNA from a cell. Ouch, that sounds like it could hurt. Well, after that happens, then the DNA is treated and it's processed. And the genes in the DNA are then identified and their nucleotides are sequenced. The last step of the process is to find the sequences of the DNA barcode region. Then this sequence can be matched to a sequence from a library of known sequences. The library contains DNA barcodes from specimens that scientists have already identified.
All right, so now it's your turn. In this lab, you're going to actually follow the first and the last steps of DNA barcoding. For the extraction step, you're going to use a strawberry instead of a fish. Now, because each strawberry cell has 8 copies of its chromosomes, you're going to actually be able to collect a large amount of DNA from your strawberry. Using a fish would be a little bit more difficult. The other thing about strawberries is that they also contain enzymes, and these enzymes will help break down those cell walls while you're doing your investigation. So go to your assignment, you should already be in it, and it has all of the instructions for you to follow to do your own DNA extraction from this strawberry.